Hi, everyone. I'm your host, Dr. Marie McNeely, and I'm delighted to welcome you to episode 593 of the People Behind the Science podcast. This week, we are excited to be rebroadcasting our interview with Dr. George Hajishengallis, originally published on December 5th, 2014. Listeners, George is conducting fascinating research on the microbes that live in our mouths and how our immune system interacts with these microbes to better understand how homeostasis is maintained and how the balance can break down to cause disease. Research from his lab has brought to light novel mechanisms of microbial dysbiosis, inflammation, and tissue homeostasis. So listeners, I hope you are ready to learn more and to be inspired by our guest George in this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. I am delighted to be here today with Dr. George Hajishengallis. So welcome to the show, George. How's everything going today?
Oh, that's fine. Today is a nice day outside, and I'm glad that we will be talking together.
Absolutely. Well, I am looking forward to our conversation, and I am excited to introduce you to our listeners. Now, George is a professor in the Department of Microbiology at the University of Pennsylvania. He received his Doctor of Dental Surgery degree from the University of Athens in Greece and his PhD in Cellular and Molecular Biology from the University of Alabama at Birmingham. He then served on the faculty at Louisiana State University Health Science Center, as well as the University of Louisville, before joining the faculty there at University of Pennsylvania. Now, George has been elected as a Fellow of the Japanese Society for the Promotion of Science and was also the recipient of the International Association for Dental Research's Distinguished Scientist Award in Oral Biology in 2012. And George is here with us today to tell us all about his journey through life and science. So today we want to get to know you, George, both as a scientist, but also as a person. So can you take a minute to tell our listeners what you like to do when you're not doing science?
Well, there is not much time for that, but I like watching soccer, reading a book before going to bed, and hiking plus swimming with my wife in summertime.
That sounds wonderful. So a soccer fan, definitely getting outside, enjoying the great outdoors, and a good book too. I like that. So George, great to hear a little bit more about how you like to spend your free time. Can you tell us next about how you spend your time at work? How do you describe what you do to someone who might not be in science?
Yes, we are trying to understand how the immune system interacts with oral bacteria and why in some people this interplay leads to inflammation that destroys the gums and the bone that support the teeth. We're also working to develop treatments that can prevent this gum disease known as periodontitis.
Certainly. And I was shocked to find that nearly half of adults in America have periodontitis.
Yeah,
that is a staggering statistic there.
Yes, 47% have some form of periodontal disease, and severe forms probably apply to 10 to 15% of the people. And in its severe form, periodontitis can adversely affect also systemic health. People with periodontitis, they run higher risk for developing atherosclerosis, diabetes, adverse pregnancy outcomes, and several other systemic diseases.
So a very important area of study, and I look forward to chatting a little bit more about some of these details of the projects you're working on later in the interview. But I want to chat now about what motivates you, George. I love to ask scientists if they have a favorite motivational quote or a saying that really resonates with them, that helps them get through each day. So can you share yours with us today?
Yes. Chance favors the prepared mind, which is attributed to Louis Pasteur. Though a nice quote, it does not sound like motivational, but it is because it motivates you to be inquisitive, to think laterally, and be ready when luck strikes.
Absolutely. Well, I think that is a great quote and a great reminder to keep with you, as you're going through your day as a scientist, to always be prepared, to kind of do your homework, do the work ahead of time, and then be ready to take advantage of those chances and opportunities. I love it. So can you tell us a little bit more now about some of these people who may have motivated you or inspired you along your journey, some of your role models?
Before my PhD in the States, I studied dentistry at the University of Athens in Greece, as you mentioned in the introduction. A professor of mine there, who was both a dentist and a biochemist, inspired me that I started to appreciate that dentistry is also a science. And I wanted to learn more about the biological basis of dental diseases, more than was written at the time in the textbooks. And that is why I decided to pursue a PhD in a basic biomedical science. Also, throughout the years, I have been fascinated by the ancient Greek philosophers, not for the answers they provided, but for the questions they had asked. They were the first to attempt to give a purely natural explanation of the origin of the world, free of any mythological connotations. That was the first time this happened in history.
I think that is great to draw inspiration from these brilliant Greek philosophers who were really starting to take this logical scientific approach to describing the natural world. I think that's fascinating. And certainly the mentor in your early days as a career is, one of these people that a lot of scientists cite as someone who really had a big impact on their life and on their career. So great to hear about how you get your inspiration and some of these people who've inspired you. Now, I'm curious to hear, George, about the early days. So can you take us back to the very beginning of your scientific interest or scientific career and tell us what got you excited about science and maybe what directed you towards this particular field in dentistry?
Sure. Like I said earlier, I went to dental school, and although I liked dentistry, at some point before I graduated, I was afraid that dental practice would become routine and I would lose the stimulus of constant learning and questioning things. That's when I decided to come to the United States and pursue a PhD. And I earned a PhD in Cellular and Molecular Biology at the University of Alabama in Birmingham, and then did a postdoc in Immunology at the same university and also at the State University of New York at Buffalo. Then I had faculty appointments at the Louisiana State University, the University of Louisville, and joined the University of Pennsylvania in 2012, where I'm now a professor of microbiology, as you mentioned, at the School of Dental Medicine. And this is in a nutshell about my career, how it started and how it's developing now.
So was this always your dream to have this career in dentistry? Or maybe when you were a kid, did you have other careers that you were considering or other paths that you thought about taking?
I thought about becoming a chemical engineer because I liked chemistry, but then I realized that chemical engineering was more about engineering than about chemistry. So I found in biology something even more exciting than in pure chemistry, finally.
So you didn't have that maybe inspirational dentist as a child that you wanted to follow in the footsteps of? Very interesting. Well, George, great to hear about your career path and really how you got to where you are today, and certainly some of the other science fields that you were considering as you were making your decision. Now, can you tell us a little bit more about what you're doing now and one of these projects that you're working on that you are so excited to be researching?
Yes, I think we are doing several projects, but I will describe one that I think was quite exciting, and we just published a study recently, and we are still continuing on it. So although periodontitis typically affects adults, there are certain cases where young children develop aggressive forms of this disease. For instance, in kids with a genetic syndrome called leukocyte adhesion deficiency, or LAD, gum inflammation may completely destroy the supporting bone and the kids will lose their teeth. This will happen even if the children receive antibiotic treatment and professional dental care. Now, in patients with LAD, what happens is that the white blood cells, like the neutrophils, lack certain adhesion molecules called beta-2 integrins, and the neutrophils cannot migrate from the blood vessels to the tissues like the gums. Now, historically, scientists thought that the etiology of these conditions had to do with the inability to handle the bacteria in the gum tissue. That notion was very commonsensical. If you do not have neutrophils, this type of white blood cells, you probably cannot control gum infection. Mm-hmm. However, we have shown in a recent paper that was published in Science Translational Medicine that this time-honored idea was wrong. We have shown, in collaboration with dental scientists from the NIH, that the lack of neutrophils in the gum tissue causes dysregulation of the host immune response. Specifically, there is overproduction of a pro-inflammatory molecule, which is called interleukin-17, and this is responsible for the bone loss. We're currently trying to understand more about this mechanism, but what can be said at this time is that blocking interleukin 17 in these patients is a promising strategy to prevent them from losing their teeth. This strategy has been tested by us and has worked in mouse models of the disease, and it is something that will be pursued in collaboration with NIH, which has access to this rare disease.
That is a fascinating project. Now, we're talking about this aggressive form of periodontitis in children. Is this something where, they may lose their teeth, but are these just their baby teeth? Does this affect their permanent?
Both dentitions.
Okay.
It affects both the primary and the permanent dentition, unfortunately.
Interesting. Well, that is fascinating disease. And I think interesting to think about, the sort of, juxtaposition, thinking about in adults versus this aggressive form in children. So how quickly of a time period are we talking between when it starts to be a problem in a child and when they start losing teeth?
I have not seen directly the patients. We have done the basic work and the animal model work. Our collaborators at the NIH see the patients. But I have seen pictures of some very young people, children, like 11-year-old boy with having lost half of his teeth. And I know that this disease starts at very, very young age. So they start losing bone when they are really very young. And like I said, even when they are 6-7 years old, they start losing teeth, which is not only a functional issue, it's also psychological for these kids. So we're talking about a very severe form of periodontal disease.
Certainly. So visually, can you distinguish this very rare, relatively more aggressive type of periodontal disease from sort of the standard run-of-the-mill periodontal disease?
Well, the fact that it occurs in young children is enough to say that this is not the typical form.
Okay.
The typical form affects adults. Also, the histopathology shows a very severe infiltration by inflammatory cells. And you can see all the players there that you would see in adult forms. But like I said, there are no neutrophils there. So this is quite pathognomonic. It's a nice way to distinguish the two diseases. There are no neutrophils in the gum tissue, whereas the blood vessels are full of neutrophils that, of course, cannot get out to the tissue. They cannot transmigrate, that is.
Well, this is a fascinating research project and certainly very important, like you said, not only affecting physical health and these children, but also the psychological impacts as well, losing teeth that early and not having them come back. I think that is certainly a problem, and great to hear that you're able to make some strides in this research line. Now, I think so far, George, we've talked about a lot of the great aspects of your career and your journey as a scientist and some of the fascinating research that you're working on. And we haven't yet chatted about some of the struggles or some of the challenges that you as a scientist face. And I think every scientist can look back on their career and there are these moments or periods, maybe days, maybe weeks, maybe even longer, where it just seems like everything is going wrong and there's just major struggles or maybe a major failure that happens. And I think it's very important to talk about these because I think in the literature, in the media, you see all the success stories, but you don't hear about everything that came before it. So can you take us back, George, and tell us about one of these times of struggle or failure in your career, and what are some of these steps that you took to get yourself through it?
Well, getting my first grant, I was naive enough to not think about the consequences, that is losing my job, and was absorbed in what I thought was an exciting science. So I cannot say that I felt any fear of failing, although I should have.
Right. Hindsight right there. Interesting. So what are some steps that you took to get yourself out of this difficult situation where you were trying to apply for that grant, things weren't coming through? What did you do next?
Like I said, I was so naive that I thought that what I was doing was interesting, regardless of whether, the reviewers appreciate from the beginning. So I just continued doing more experiments, rewriting it. And like I said, I was absorbed in the science itself, so I was not worrying about it. So I didn't realize that would happen. And eventually I got the first grant. And once I got the first one, the rest was quite easy. I mean, when I say it's easy, it takes a lot of effort.
Right. Easy is relative.
But like I said, I like what I'm doing. And the excitement I get from doing science helps me overcome any negative thoughts or, possibilities for failure and things like that. So I cannot say that it has been a stressful life in that regard.
Well, that is wonderful to hear. And I think I love this approach that you've taken, sort of being so immersed and excited about the research that, those negative reviews kind of roll right off. So what would you say, when you get this email perhaps or letter, what was your response the first time that you saw that first rejection on a grant? How did you feel? What were you thinking?
Well, it was not an exciting news. You feel disappointed. But fortunately, I have this negative feeling only the first day. And next morning, it's another day, and you start doing what you had in mind, and you don't care about the negative review. You hope that the next time will be better. It took me 3 times the first time. I sent that grant 3 times over a period of 2 years before I get the first grant. But like I said, I liked what I was doing, so I was not thinking about the possible negative consequences that this would have.
Certainly. Well, I love that approach. Give yourself a day to be sad and disappointed about it, and then go in the next day ready to go, optimistic, and starting fresh. I think that's fantastic. And, I think that is such a great, healthy way to approach a lot of these failures that you encounter in science, because like you mentioned, getting these grants is a struggle. Oftentimes getting papers published is a struggle. Like you said, 2 years before you were able to secure that grant, and that's normal,
Yes.
So fantastic to hear how you cope with some of these challenges that scientists face. But we don't just want to talk about the tough times, George. Can you tell us next about a success story, one of these occasions, and it can be a big success like a grant or a paper, or even one of these small victories along the way in your lab or in your research?
Well, when we talk about success in science, we usually mean grants, awards from scientific societies or good publications. And I have been fortunate to have these kinds of success, but I think success has more to do with good work and its implications. Translating science, for example, is very important, and we have recently shown that blocking complement, a system of innate immunity involved in inflammation, prevents the development of periodontitis in non-human primates. This is the closest model you can have for human periodontitis. Now, in collaboration with the medical school at the University of Pennsylvania, we are very interested to pursue human clinical trials. Now, if that works, we get there and we treat humans and we prevent disease there. That will be the biggest success I could imagine for me. Now, in terms of basic concepts, I was excited that we have recently helped develop the keystone pathogen hypothesis for the etiology of periodontal diseases. Until recently, a few select bacteria, such as Porphyromonas gingivalis, were considered causative agents in periodontitis. We have shown, however, that Porphyromonas gingivalis actually cannot cause periodontitis by itself. But what it does is to convert a previously commensal or benign microbial community into a pathogenic microbial community. We call P. gingivalis, short for Porphyromonas gingivalis, a keystone pathogen. That is, a pathogen that is with a disproportionately large effect on its environment relative to its abundance. Now, this is a metaphor in analogy with the literal keystone. The literal keystone is only one of many stones in an arch, but it's critical for the overall structure. In the same way, P. gingivalis is very critical for the microbial community to convert it from a commensal pathogenic state. So this is how this pathogen mediates disease. It's not that it can directly cause disease, which was thought until recently.
Interesting. So in cases in normal, healthy gum tissue or oral conditions, you have this P. gingivalis, but it's not causing problems?
This is a great question. Well, first of all, there is heterogeneity within the population of P. gingivalis. Not all strains are equally pathogenic. So that's one possibility. The other possibility is that even though Porphyromonas gingivalis may be present in some people, certain people, because of the intrinsic immunoinflammatory status, can either resist or tolerate the conversion of a commensal microbial community into a pathogenic community. For example, people may have gene polymorphisms or gene mutations that attenuate the inflammatory response of the host, or they may have mutations in critical receptors that are required by the periodontal bacteria to evade or subvert the host response. So, like many other chronic diseases, periodontitis is also of multifactorial etiology. Whether somebody develops periodontal disease can be determined at 3 different levels. At the microbial level, that is, what is the composition of the bacteria that populate the periodontal tissues of each person. At the host genetic level, The genotype may determine whether somebody develops hyperinflammatory or destructive responses or not. And also very important at the environmental exposure level. I mean, smoking, for example, has been shown unequivocally to increase the risk of periodontitis.
Really?
So environmental factors can determine the outcome of periodontitis either in a positive or a negative way. So it's a multifactorial disease, and bacteria are necessary. I mean, if we lived in a germ-free environment, nobody would have periodontitis. So
right, we'd be in trouble.
Yes, but they are not sufficient by themselves. It's actually the host response to this microbial challenge that mediates the inflammatory destruction of the periodontal tissues.
Interesting. Well, that is a fascinating project, and it sounds like you've had some exciting success in your research, and certainly this sort of big one on the horizon with the complement, moving into humans potentially. I think that is fantastic. So do you have a favorite way, George, to celebrate some of these research successes in the lab?
Well, when we have some success, we may go out for dinner and celebrate it. But I think the greatest excitement, at least personally, I get, I have it when I first see the first data from students or postdocs. Writing it is also exciting, but it's not as exciting as the first time that you see the raw data and you understand that you have something really exciting there.
Absolutely. I think, it's hard to contain your excitement while you're writing it, It seems like a long process, and you're just so excited to get that information out, to share it with everybody, because you are one of the few people in the world that know that at that point. I think that is fantastic, and certainly one of the wonderful aspects about careers in science. So great to hear all these success stories in your lab. And I think researchers keep so busy with the research, with keeping up with the literature, publishing papers, things like that. It doesn't always leave a ton of free time to read. But you mentioned you enjoy relaxing with a good book when we were chatting in our introduction there.
Yes, I read every day about 10 or 20 minutes before going to bed. Actually, even exciting books, they help me go to sleep easier.
Really?
Yes. But yeah, I enjoy reading a book, but that's the only time I have for reading books, just before bed. During the day, there is no time for that.
Certainly.
Even during weekends, I don't have time other than bedtime.
Well, it's good to reserve those, last 10, 20 minutes before you fall asleep to relax and enjoy a good book. Now, can you tell us about one of these books that you've read that you loved and would recommend that our listeners check out? And this can be a science book or a non-science book.
Yes. Many years ago, I happened to read a book by David Lodge. That was a hilarious book satirizing academic life. And the title is Changing Places, A Tale of Two Campuses. More recently, the one that I'm reading now, I just started reading the history of the Peloponnesian War, which is written by Thucydides, an Athenian historian who also served as an Athenian general during that war between Athens and Sparta. And perhaps this is the earliest scholarly work of history, and Thucydides did not trust information that was not experienced firsthand or was not confirmed independently. Now, I was taught this book when I was a high school student, actually in ancient Greek. But rereading it after many years, you can see things in a different perspective. And I can enjoy it more now that it's up to me where and when to read it.
Right.
To take it as a class.
Certainly. I think I found that with a lot of the books I read in high school. I didn't like them at the time, but when you pick them up later, you're like, this is actually a really good book, now that I don't have to have chapter 3 read by tomorrow.
Exactly.
So. Well, that is fantastic. So do you find yourself reaching for a lot of these history books, or do you like a variety of different kinds?
Variety of different kinds. I like popular science books. I like, for example, physics, astronomy. I have read several over the years. Historical books I like. Fiction books sometimes, yes, like Agatha Christie and things like that. Mm-hmm. So a variety of books.
Fantastic.
I try not to read science. So.
Yeah. Yeah, you like to at least get outside of your own field in science when you're reading for fun.
Yes.
Well, that is fantastic, George. Great to hear your book recommendations. And we'll put those 2 books that you mentioned out on our website for our listeners to check out. And now I know one of the ways that I'm able to squeeze in a little reading in addition to right before bed is when I'm traveling. So sitting on a plane, reading a book. And I know scientists actually get quite a few opportunities to get out there and travel to share their research. And this might be for conferences, working with collaborators, things like that. So can you think back now, George, on all these places that you have traveled for science, and tell us about your favorite and just what about it made it so memorable?
Yes, the island of Crete in Greece. It's a fantastic place with unique culture and landscape, beautiful beaches, and really breathtaking mountains and gorges. I have attended several international conferences there, some of which I helped organize. For people who know me, they will think I have been biased saying this to you.
I was going to say, is this where you grew up?
No. My wife comes from Crete.
Okay.
But I'm not saying this because it's the place that my wife was born. It's really an exciting place. I mean, anybody who goes there will attest to what I have said.
Wonderful. So what are some of the sites that we have to see when we're in Crete?
First of all, the San Maria Gorge, it's a must-do thing for most of the tourists. So it's a gorgeous gorge, and it takes 5 or 6 hours for most people to walk through it. And once you arrive at the other end, the sea in the southern part of the island, so you can swim there. You don't have to walk back, but you can take the boats and go to another place, and then you can take a bus to go to where you started from the north part of the island. The food there is unique. I mean, anywhere you can go, you can have great food. San Maria Gorge is only one of many gorges that you can hike. It's a wonderful place. I mean, there are also archaeological sites that are unique there, like in Knossos. This is probably the center of the first European civilization which has been developed in Crete in 3,000 or 2,000 years BC. So there are plenty of things to do and see there.
Wonderful. Well, this gorge excursion sounds fantastic. You mentioned you like hiking and swimming, so that's the best of both worlds there.
Yes, exactly.
I like that. Well, it sounds beautiful the way that you're describing it. So great to hear about this place that you've been able to go to for a couple different conferences, certainly organizing them there as well. And I think it is a nice way to get out, see the world, and also enjoy some great conversations about science and meet some wonderful scientists. And I think when scientists look back over the course of their careers and all these people that they've worked with in the lab, all these people they've collaborated with, it's amazing. And you form these bonds. And I think when you look back at these different lab groups and research groups, they have their own personalities. And contrary to some of these stereotypes about scientists, I think a lot of the people are just amazing and creative and oftentimes quite funny. And with all these brilliant people working together, you get a lot of quirky traditions that spring up and even just some funny memories that you can look back and laugh about. So can you share with us today, George, one of these traditions or one of these memories of yours?
Well, when I was a graduate student, I had a mouse immunization experiment, but I and lab colleagues noticed that a poor mouse that was constantly bitten by other mice. So we removed it from the study and put it into an individual cage. In other words, we elevated it to a pet status. Mm-hmm. We called him Larry.
Oh, Larry.
Vaccinated him with whatever experimental vaccines the lab had. And actually, it lived to a very old age. It was very fun. I mean, I don't remember any details after so many years, and it may sound silly, but trust me, the lab colleagues there, we had a great time with Larry. It was our pet. It was a way, to diffuse your stress, say something, to divert from everyday's work. So this is something that actually, when I thought about funny things that happen in the lab, this is the first thing that comes in mind, because I have to say that most of the time, maybe I have been more serious than I should when I was doing lab work. Of course, now I'm not doing lab work. I only write papers and grants, and only the students and the postdocs in the lab do the experimental work. But from the time that I was doing research myself, this is the thing that I remembered, Larry, the mouse that was removed from the study because it was constantly beaten by...
Everyone was picking on him, yeah. Well, that is an amazing memory, George. And I think certainly you're absolutely right. Scientists, we take the science and the research so seriously, and there's always so much work to do that, it's hard to squeeze in some fun time, but it's nice to have that great environment when you have, one of these memories like a little lab pet or just something that brings everyone together. So did you guys have to appoint people to take care of Larry, or who has to clean his cage this week or things like that?
Well, I just remember that one day I saw Larry in a huge cage that actually was intended for rats, not for mice. And I asked one of my colleagues there, I even remember his name, Terry. I said, Terry, why Larry is in this huge cage today? He said, no, don't call him Larry. This is Cell Lawrence.
He is in his castle today. Yeah.
Yes. Has been elevated to Cellular Lawrence, so this has to come with the appropriate residence.
Well, that is amazing. I love it. Well, that sounds like a fantastic memory, and great that you were able to really, I think, save this mouse from a difficult situation that he was in, being picked on by all the other study mates, and now he had a good life.
I mean, the standard procedure would be to sacrifice it and remove it from the study, of course. That would be inhumane to leave it there. But I think we did something even more humane than sacrificing it. So that goes outside the protocol.
Right. Shh, we won't tell anyone. Sir Lawrence was a fantastic experiment of his own there in being a pet. Well, that is a wonderful memory. And I love hearing about these different quirky things and memories that scientists have that really brought the lab together, made it a fun place to work. And this sounds like a great example. And in addition to some of these research questions that we've chatted about today, and we've talked about some of the work that you're doing, some of the work that your lab has done over the course of the years, I love talking to scientists about their dream research questions. And I think oftentimes scientists point to things that are standing in the way of them being able to answer those questions. And this might be funding or staff or technology or feasibility, all these normal research constraints. So if we took all of these constraints away, George, can you tell us what is one research question that you are just dying to know the answer to?
Yes, what I have in mind cannot be done, but since the question is hypothetical, I will answer hypothetically.
Sure.
So the keystone pathogen hypothesis I mentioned earlier was developed based on our work in animal models of periodontitis. I wish it were possible to test the same hypothesis directly in humans. I believe that implanting Gingivalis in the mouth of people who do not harbor this bacterium could confirm the hypothesis. Of course, it is impossible to get approval for such study, nor would I like to pursue it in this way. It's not ethical, but it would be something that I would like to know the answer.
Absolutely. Well, when you're talking about human studies research, a lot of this health type research, you run into this constraint a lot, to design the perfect scientific, study design.
That's why they're using animal models.
Exactly.
I know that animal models have been criticized, that they do not faithfully represent all stages or all aspects of a given human disease. But my answer to that is that's why we call them models. And the models should not be evaluated on the basis whether they faithfully represent all stages of the human disease, but a model is to address specific hypotheses. So that's how models should be used, answer different questions each time.
Absolutely. And you can always ask those people who are criticizing them, would you like to be a human study in this subject?
Or would you like to have your kid taking a drug that has not been tested in animals before?
Right. Well, I think this is an interesting question, and certainly pointing to some of the, limitations in doing these questions that you hope to ultimately apply to humans. But, it's really difficult to get through this process. You have to work in the animal models and then sort of elevate it up the chain until you're able to safely test them and do those experiments in humans. So definitely one of these things to think about when going into this field. Now, I love chatting about those research questions that you're not yet able to answer. And I love talking also about some of the advice that you've received over the years. I mean, we talked about certain people who you've worked with and that mentor that you had in your early years. Can you share with us today, George, what you would consider the best piece of advice that you've ever received in your career or in your life?
Yes, something that I heard in high school. So the advice was something like that. Take your time before you decide, but once you make up your mind, act promptly. Now, in the context of science, in other words, that would mean take your time to develop a good idea, but when you have it, convert it into action swiftly. Because ideas without experiments do not help you in science.
Certainly. Well, I think that is great advice. And I love really focusing on taking the time, developing those ideas, developing the plan. But then once you have it, you have to go,
Yes.
And I think in different fields of science, that sense of urgency is stronger because you might be trying to get your information out there before another group thinks of that same brilliant idea and gets it out there. So certainly great advice to turn those brilliant ideas into actions, into experiments, and get the data out there. Now, is there any other last piece of advice, George, or maybe last piece of inspiration that you want to share with our listeners now, coming from you personally?
Yes, I think I alluded to it as I was speaking. So I would like to say, or suggests that the excitement is in the journey, not in the glory. And this is derived from Ithaca, a poem by Constantine Cavafy, in which the poet implies that the enjoyment of the journey matters more than the destination itself. In other words, when this applied to science, I would like to say that if you do not like the process of discovery, but you only like success, then it may not be a good idea to pursue science as a career. So this is what advice I would give. And another thing that I would like to add, and this is completely personal, nobody has to take it and it may not work the same with every people, is that never compete with anyone else than yourself. Today, try to do something better than you did yesterday. Of course, somebody might say, we live in a competitive world. But I believe still, if you focus on what you do and try to improve yourself without caring if the others are better or worse than you, you can still become competitive without taking unwarranted pressure.
Absolutely. Well, George, I think those are two fantastic pieces of advice. I mean, I think in science, you spend so much more time on the journey, on the discovery part of it than reveling in success. So certainly, if you're one of these people that's not motivated by the everyday going out there, making discoveries, doing the work, you're going to be disappointed a lot of the time. And then certainly, I love this piece of advice about not comparing yourself to others. And I think in science, it's difficult because especially in your early days in your career, you kind of look around at everyone and you're marveling at how everyone is able to achieve success, get these publications, get these grants. But you're definitely right there. You need to focus on doing better yourself every day and really becoming the best that you can be rather than comparing yourself to everybody else.
Let the others compare yourself with others. Don't do it yourself.
That's right. Well, that is great advice for our listeners out there. Now, can you tell them, George, how they can reach out to learn more about you or your research?
The best way is through email. I'm always reachable by email, even when I'm traveling. And my email address is pretty simple. It's geoh@upenn.edu.
Fantastic. Well, listeners, definitely reach out to George if you have any questions. And George, thank you so much for joining us today and sharing your story.
Thank you very much for having me. I enjoyed our discussion and the questions were very exciting for me to answer.
Well, thank you. It has been a pleasure for me as well. And thank you to our listeners out there. Join us again next time for another episode of People Behind the Science. Your voyage to the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to seeing you next time on People Behind the Science.